Welcome, everyone. Thank you for joining us for today's webinar, Biologics Tech Transfer and Validation at Scale. I'm Heather Masaccio, your host from Outsourced Pharma. I have just a bit of housekeeping before we get started. If you have a question for our speaker or wish to share any thoughts of your own, please submit it through the Q&A box on your screen. Our speaker will address those questions at the end of the presentation today. Now, if you run into any problems with the stream today, please try refreshing your browser first. If that problem persists, go ahead and use that Q&A tool to submit a question, and I'll help get you back on track. This event will be available on demand. You'll receive a link via email as soon as it's available. Now on to our speaker. Today, we are joined by Richieri, Chief Operations Officer at Avid Bioservices. I'm going to turn it over to Richard to get us started. Richard?
Okay, thank you very much. Today's talk will be about the biologics tech transfer and validation at scale. Right now, I'm Richieri, the Chief Operations Officer at Avid. I've been at Avid a total of 20 years, with the last 6 as COO. I've been in the industry for about 35 years, all in cell culture, GMP. So today we'll be talking about how we tech transfer projects in, and also for those projects, how we move on to process validation. So just a little bit of background about Avid, just so you see the company we are, of our experience. We have over 600 batches of clinical, about over 275 batches of commercial, many years of experience. We are a completely single-use facility, so all the processes that I'll be talking about today is single-use from beginning to end. There's no stainless steel, no cleaning up parts, no sterilizations involved. We've had many years of successful manufacturing, both clinical and commercial. We have 7 commercial projects right now, with more on the way. And we're really proud of our regulatory record over the last 15 years, 043s from the FDA. So we've been inspected many times. And in fact, for the last 2 commercial products, we haven't needed the FDA did not require to be on site for a PLI. So quality is our main focus here at Avid. And under that quality umbrella, we perform these tech transfers and process validations. Just a little bit of, just so you see where we're at, we are in Tustin, California, which is essentially between Los Angeles and San Diego. We have 2 facilities. The main facility, the GMP facility in Tustin, I'll touch upon in a minute. That's where we do our commercial and PPQ work. And we're opening up a new early phase facility about 5 miles down the road in Costa Mesa, where clients will come in for early phase, where the point of this facility is to go from sequence to an IND within 40 weeks. So we want to go speed is of the essence for the Center of Excellence in Costa Mesa. But whether a client comes into Costa Mesa and then we moves on to GMP manufacturing at large scale in Tustin, or if a client directly transfers to Tustin for commercial, the process is the same. The tech transfer and the evaluations are the same to get the process validation. And the final slide as a backgrounder is just showing you the reactors that we have in our 2 sites. So on the left side is the early phase sites where we go up to 500 liters. And on the right side is our commercial facility with 2 production lines. One line housing 2,000 liters and our larger 6-pack of 2,000s and our newest line. Both lines have been commercially approved. So most of the projects we talk about are going into the larger 6-pack of 2,000 liters. So I'll get on to the process validation and our approach and the tools that we use to onboard projects. So essentially, it starts out with the very important part about process transfer, tech transfer. And this is where I think a lot of clients and CDMOs, they try to rush through this a bit. And this is a very important part of the program. Getting this wrong will set you back a long time. So we have a very standard approach here that has been worked time and time again, that we're very focused about. But it all starts with, from a client, from the donor site, gathering as much information as possible about the process, about the analytics, and especially about the raw materials, the vendors of the raw materials and the quality of the raw materials. Ideally, as much process information at both scales, large scales and research scales, bench scales, would be tantamount, would be really critical at this stage. And from that information transfer, you can really plan your tech transfer program at Avid, at a CDMO. So in general, you would start out with a tech transfer run just to make sure that you're using the information that you gathered. You create development runs in PD. If those look well, you do a final verification, a final pilot, and then onto the process transfer, the GMP. There's a very high level overview of the process. There's a lot of details that go in. But in general, throughout all this program, you want to make sure that you're aligned with your systems, both in bench scales and large scales and mid scales. And we'll get to that in a minute. But a lot of the times what we'd like to do here is just to make sure we avoid any questions about using different reactors or different columns or different chromatography skids. So making sure everything is the same during this process is really critical. But through this evaluation, you really want to have a chance to check and verify information going from one step to another. So technically, sometimes people want to rush and go on to the next study before the previous study is over, or before the previous study data is available. It's critical to know during this time to take a little bit of time and do some thorough evaluations of the data, both analytically and process-wise, before you go on to the next step. Because again, going backwards will just take a lot of time and effort. So you want to get it right the first time, essentially. But at the end of the day, you do have a process lock for a GMP process. This is not the validated process. This is just getting into a GMP at scale run. It's acceptable for clinical use, acceptable for human use, but your process is not validated at this point, but the bones of the process is available. The bones of the process is known at large scale. So really quickly, one of the things that really helps in the tech transfer program is making sure that the alignment from small scale to large scale is there. In this slide, we're showing the upstream equipment from both process development and GMP. At Avid, we take a lot of care to make sure not only we're matching the equipment and the make and the model, and we make sure that the software is the same, the user interface is the same, especially between our pilot 100-liter and our larger reactors. Everything is the same. And then in our smaller scales, whether it's the amber or the 2 liters, we have verified, and we'll talk about this later, we have verified a scale down model. Those 2 smaller units are a good, appropriate scale down model of the larger 2000s that we'll do the process qualification in. And then in a similar way, same program, same strategy for downstream, matching your downstream development work, matching your downstream equipment to your large scale upstream. In this case, we use ACTAs all throughout the process. So from the smallest scales to the largest scale, same software version, everything is the exact same. Typically, column chromatography is a little bit more straightforward to scale down than reactors. But again, keeping everything the same just avoids any sort of issues during that tech transfer program. And that's the strategy that we want to avoid. You want to avoid any questions, anything that could go wrong. Really try to eliminate those risks of anything that is different between scales. And in a similar way, using the same strategy for assays, which is many times overlooked during the tech transfer program. But aligning as best you can with the donor site and their methods, and then also at our site at Avid, making sure the methods used in the analytical development lab when they do feasibility studies and qualification studies and validation studies for the assays, that the development lab is the exact same version as in the QC lab. So when the processes are transferred, when the assays are transferred, it's really seamless. It is really one-to-one, like-for-like assays. So that's how you get that reproducibility from batch after batch after batch when you do this type of strategy. So just talking, so far I've been talking about the tech transfer. And whether you do an early phase or late phase or a PPQ program, it always starts with a tech transfer. There have been times where people say, let's start right away with PPQ. Let's say the first batch is a PPQ batch, and it truly can't really work that way. That first batch really needs to be that tech transfer batch. You do not want to start your PPQ using, for your very first batch. When you do your PPQs, you want to avoid any risk. You really shouldn't be anything. There should be nothing new to discover during your PPQ. So that tech transfer, that robust tech transfer that we just talked about is really one of the key attributes to having a successful PPQ program. But once the process has been tech transferred to the site, to our site, there is a program to go forward from there. You say, okay, we're going to go into PPQ. We want to validate this process. And there's generally 5 stages to this type of program in a general sense. And the first stages should be covered with the tech transfer program, where you get that initial product and process understanding. You know about your protein, you know its stability, you know how it behaves at different stages of the process, from each stage of the purification process, for example. You have an understanding of the performance of cell culture and yields in the purification. It's not validated yet, but you have a good understanding, or you feel very good about the process at this point. You feel that it's ready for PPQ. You don't see any red flags when you did your GMP that says, okay, we have to further develop. And during that tech transfer, it's very important that we qualify a scale down model. You don't want to do that during your first PPQ. You want to do that during your first GMP or your GMP prior to your PPQs. And so for scale down models, it simply means taking samples from the at scale process, whether it's a reactor or a purification column, transferring that into the labs for small scale versions of that process, whether it's a reactor or a small column. Now that you qualify that column, then the real work begins with process characterization, where you really hone in on any sort of risk that you can determine for the process. You execute on stage 4 into the process validation, the at-scale PPQ campaign, which if you did your work right at this point, it should be very seamless, no surprises. And then of course, once your PPQ campaign is done and the reports are done, the work isn't over. When you're in commercial manufacturing, you always monitor your process and your process performance and the lot release performance, and you continually monitor, and there's ways to verify and trend the data that we'll talk about. So very quickly, to do a proper scale down model, again, we touched upon this upon the tech transfer that you want to have. But again, you want to match your manufacturing scale, and you take samples from each to your bench scale in the PD labs, where you can then do your studies. Your studies are based upon risk assessments. Your studies are based upon making sure that your scaling procedures are appropriate, your engineering procedures to scale down reactors and scale down columns. You're making sure that the performance, the titer, the growth, in this case for the cell cultures, is matching your large scale. Your metabolite profiles, your CO2, your ammonia, all that is very important for product quality. And you want to make sure that your scale down model is an appropriate model for your larger scale. So typically during PPQs, your samples will be taken from large scale, as you can see in those arrows, and going into the small scale work. And in a similar vein, the samples from large-scale manufacturing for purification will then be transferred to the lab for a small-scale verification, small-scale modeling qualification. Like I said before, it's a little bit more straightforward. Columns are a little bit easier to scale down, where essentially you're keeping the column height the same, you're keeping the residence time the same in the small scale. And you really are showing that the yields per column, the product-related impurities are removed as you're intending them to remove. Your process-related impurities like host cell proteins and residual DNA are also removed. Your virus filters, your filters are also performing as intended when you scale them down properly. This is really more, again, straightforward, but you really want to challenge the limits of these parameters for each step. And just go through a very simple way to do some of these scale down models. And a lot of times you can't check every parameter for everything, essentially. So you really need to think about your process and really think about your critical quality attributes and what could affect it. And then once you see what affects it, then you really hone in on your study plan. And for example, in this case, I'm just listing some critical quality attributes here: glycosylation, aggregation, all that. But if you take a look at your critical quality attributes and you take a look at a typical process on the right, you can try to see, okay, which one of those quality attributes is affected by each of those steps. And in a very simple example, you can say aggregation is a critical quality attribute, of course. Well, where would that really happen? And for many proteins, it could happen during virus inactivation. In a virus inactivation procedure, you typically lower your pool of product after the initial capture step to a lower pH 3.5 or so, and you keep it there for a certain amount of time, 30 minutes, an hour, just to inactivate viruses. This is a very important program for virus validation. You need to show this. And this is where it's a potential where your protein could aggregate. But in general, with your process knowledge by this time, you could probably rule out items such as temperature or agitation rate during this step as causing aggregation. And you're doing your risk assessments, and then you're really looking at even more. Does adding acid and base, does that really cause the aggregation? Is that a critical factor? And then it turns out at the end of the day, it's really kind of obvious in this case, but your low pH endpoint or the whole time you're holding pH, those are the critical factors that really cause aggregation. Those are the ones you want to really challenge during your scale down. So you really stretch the limits of those two items. Instead of looking at all 7 items here, you're really looking at those 2 items in a very detailed manner. And then you're really confident when you do things at larger scale, or if there's even an excursion at larger scale, that you're covered. So that's part of process validation, is not just covering your set parameters, your normal operating parameters, but any excursion that happens, you have the data set to support any of those discrepancies. Oh, went too far. So at the end of the day, we've tech transferred the process. We've done the scale down model. We've challenged the process from beginning to end, from thaw to drug substance at the small scale with all these parameter assessments and really verifying the critical quality attributes. And now you're starting to get ready to go into the large scale. And at this point, it's really taking all this information from PC data, from product quality data, from donor site reports, any sort of risk assessments that you have, and you write your process control strategy. This is the document that really starts the ball rolling. From this document, you then create your batch record revisions for your PPQ. And of course, you need to write separate PPQ protocols that have pre-approved, predetermined acceptance criteria for each of the steps. There's typically a PPQ protocol for upstream for the cell culture part, and a PPQ protocol for downstream that goes typically from the harvest to the drug substance, those 2 protocols. And within those protocols are extensive sampling plans. We will be sampling these PPQ runs. Typically there are 3. We'll be doing an extensive sampling, more so than you would ever do for a general GMP batch. So typically after each step, you would be testing your product in a very thorough manner. So once your process control strategy is developed and approved, you can then start approving your batch records and protocols and your sampling plan, and you're ready to go. And here, at the end of the day, you do 3 large-scale runs, in this case showing 2,000-liter runs. At the end of the day, you did it properly up to this point. This is actually the least stress part of the whole program if you did it properly. It's just executing the runs and the sampling plan and gathering the data. There should be 0 worry. There should be 0 concern that you're going to discover something new at this point. So if you did things properly up to this point, so you're doing 3 runs in a row, you're showing that the manufacturing batch records, if they're executed properly, will give you the desired performance in yield, in removal of impurities, and of course into product quality. Above all is of course the safety profile that we need to maintain during all this to show that the FDA's concern, of course, is safety, and of course all this is part of the PPQ program. But at the end of the day, you do these 3 runs and you show the performance in a wrap-up report. Concurrently to the large-scale run, there are side studies that need to be performed. And these, you can leverage a lot of the studies from the donor site, from a client. For example, virus validation studies. If the purification process has not changed, there is no need to change the virus validation. There's no need to repeat the virus validation studies or column lifetime studies or cycling studies. If the process hasn't changed, there's no reason to change those studies. So there's a lot of knowledge from the donor site that could be used to reduce the need for a lot of these side studies. But there's always some side studies that you need to do. And I showed a few here as some examples. So for Upstream, the LIFCA and EOPC studies are typically done at a PPQ stage. That's where you want to show that your cell line is stable for many generations beyond a typical batch. And in downstream, of course, a very simple study, but a very important study is showing impurity clearance and mapping the impurities as you do your purification. Typically, your purification consists of 3 to 4 columns or other types of steps. And you want to show how each step is removing, or if they are removing, some of these process and product related impurities. So going back to the EOPC stability data, it's very simple data, simple experiment, if you will, or study. What you like to do in one of your PPQ batches is to cut back the cells. And you can do that early in the shake flask stage or the inoculum expansion stage. And you want to do that so you're like I said, a few generations beyond what is typically done in a traditional batch. And the idea here is when you get these, at the end of the day, the batch that you made is using a cell line that has a few more generations. At the end of the batch, when the big reactor is harvested, you would then take those cells that have seen those extra generations and you send those out for stability genomics testing. So you really want to show that the genetic stability and that safety data is there, and that's a very key part of your BLA. That's a very important report that needs to go into your BLA. And then similarly, showing on the downstream side, showing the chromatography steps or removing the process-related impurities. It's written into the PPQ protocols and the batch records to take these samples after each step. These samples would typically be sent to different labs that specialized in, say, certain assays, host cell proteins, and at Avid, and other impurities, maybe at other CROs. But at Avid, we do the host cell proteins, residual DNA, residual protein, those types of removal. We can do that here. But it's important to show you have a robustness to the removal, and then you're going to meet your lot release specifications with ease. That's really the whole point here, that each stage, each purification step is doing its job in removing these impurities from the from your product that you're going to eventually fill. If turns out it's very rare, but if there's some impurities that may remain a little bit high risk, if you determine that, you can still test these things as part of your IPMS in terms of production scale at the end of the day. But typically, by this time, most of these impurities are well below any sort of concern. And just showing you the tests that people typically do at each stage. So it's showing a very simple process on the right, from protein A, post-protein A depth filter, and anion exchange and cation exchange, really taking samples from each and every one of these steps for each and every one of those purity tests. It's a lot of sampling, but at the end of the day, you have a complete profile that in the future, you don't need to test those samples every time you do a typical batch. That's the whole point. You're validating this for a reason. You have a very clear story about your program. And when you submit this in a BLA, you really don't have to worry about that if the FDA accepts this data as doing this as a routine sampling plan during your traditional commercial batch. So right now, we've done everything so far with the PPQ protocols. You wrap up that last batch, PPQ batch, you wrap up the supporting studies. They're all closed. After this, you want to start revising your control strategy based on these results. You want to revise your batch records to be the commercial batch records now. And of course, you want to close out the master report to close out the PPQ program. One of the things that's very important, like we like to do here at Avid, is that we go through all this validation for a reason. And one of the benefits of this PPQ protocols, the program, you can really now make your batch records much more streamlined than you normally would. If you validated something to be removed, if you validated a process, your checks and balance, your checks during the batch records could be reduced. They're still there, but they're not as many samples as you would normally take in a GMP batch that prior to being validated. You can really rely on your process validation program, if you did it right, to make a more simple, streamlined batch record that you're just focused on the important critical data and the data that affects your critical product attributes, your CPPs, your critical parameters. Those are the parameters you really want to focus on in your batch record, and making sure that those have the proper limits and for normal operating ranges. And then, as I mentioned before, after all this is done, the work is not done. The PPQ program is complete, submitted in a BLA, hopefully approved. And then once it's approved, going into commercial production. And then even during commercial production, while you execute the batch records, you have to always continuously verify the process. You have to assess each batch and trend each batch that I'll show in a minute. So at Avid, we have an SOP governing, it's called process verification. So essentially each batch that we do, we will verify, we will trend the data and compare it to previous batches. So you do need a good amount of batches to really make it, the data more valuable. So, but you have to start out somewhere. But in general, what you're looking for is not just that the batches are being released within spec in these commercial batches, but you're looking for trends. For example, is there a trend where you have six batches in a row that is increasing in a certain parameter or a certain assay result? It could still be within spec, but what's going on there? Why is it trending up? There's other trend evaluations you can do if it's below a certain controlled parameter for a certain number of batches, or if it goes up to if it goes up and if it goes down too much below the control limits. There's always something you need to assess. So you're looking at trends, not value, not the results, but the trends. And there's certain rules that you need to abide by, and certain rules that are published that say how to evaluate these trends and what to look out for. These don't prevent lots from being released, but they do warrant more investigation, like what is going on? Do we have a problem here? And the idea of this process verification, just to make sure as a sanity check, is there an issue going on that we don't know about? A very simple example could be if a vendor changed a raw material that caused some of these results. Did that happen? And so this is a way you can kind of check and do a sanity check on your process and your lot release data, and really challenge it and making sure you assess it and not just say, okay, it passed spec, we're good. But you really want to go to that next level of evaluation. And of course, really what we've got coming up at the end of the day here is doing it right, taking your time during tech transfer, doing the evaluations at small scale, doing a real thought process about risks really leads into successful performance, and not just successful performance, but reproducible performance. You want to see robustness, you want to see reproducibility. you don't want to see one batch out of 3 just not look as good. It could be still within spec, but not be the same. That consistency of performance, batch after batch, time after time, is really what we're looking at here for process qualification, process validation. And also, when you have a spec, a release spec, you want to make sure you're not on the edge. You know, if your SEC is 95%, you don't want to be at 95.3% as a routine result. You really want to be well above spec in every one of these lot release assays. Just a few quick examples of case studies, and just showing you really at the end of the day, these case studies are showing that the power of the scale down modeling, the power of process knowledge will really lead to successful PPQs and also a reduction in time. And the first example I'm showing is a client that came to us with the same platform as we have. In this case, it was the thermo reactors at 2 KL, same platform and scale. And we did not do the PC work. We did not do the scale down work. The client did. And they did it so well that we did not have to do any work here. And so what we're pointing out here is not just the data that looks acceptable in both cell culture and purification and lot release, but the timing. We were able to go from start to GMP thaw in 6 months. And then more critically, because the client did all this PC data, because it was very good work, we were able to go from the end of that GMP run to the start of a PPQ campaign in 3 months. By doing that work up front, by having that process knowledge, by having the normal operating parameters understood, by having acceptable ranges understood, we were able to go very quickly from GMP to PPQ. And this is again, it helps when we do it at the same scale, but again, having that qualified scale down model, that really accelerates the time and also accelerates or gives you better understanding of the process at scale, and more confident that that the PPQ will be successful. Same concept, but in this case, this is a client that had a different scale and had a different reactor platform, but they still did a great job with the small scale PC studies. And in this case, again, we're going from 0 to PPQ in a very fast manner. The scale and the performance and the product quality was known very well by the donor site, by the client. The PC studies were so robust that we're able to, transfer that process from a different type of reactor platform into our platform. We matched their KLAs, for example, or we matched their mixing and their gassing, and it was very successfully done. So it's not just matching equipment, but again, it just shows the power of doing the small scale work and a very serious PC study that allows you to go very quickly from a donor site to the final manufacturing site. And then finally, this is a client that came to us without PC data. However, and this is during the tech transfer process, and we're getting ready for PC work. But it just shows that when there isn't PC data, when there isn't small scale work, you have to at least do some robustness studies on your own. It may not be a true process characterization study, but at least do a DOE type of study that can to give you some confidence about the ranges that you're going to. So in this case, without this PC study, and we know how critical it is for this client to get to PPQ, we wanted to make sure that that first GMP run was successful. And in this case, at Avid, we did an AMBER run where we did a very high throughput screening of parameters in the AMBER system that we have. We have a 24-way AMBER. We can do 24 different conditions. And again, making sure that was good allowed us to go into GMP as quickly as practical, with confidence that the process would work. And like I said, if that tech transfer doesn't work, that really sets you back on your PPQ. So getting the tech transfer right, even if you don't have all the data, but you have enough data to feel confident that you have the basics of the process, this is probably one of the more critical things that that can happen during a program. You definitely don't want to have a setback, and you definitely don't want to reinvent the wheel, and you definitely don't want to investigate what went wrong. We want to make sure that that tech transfer goes well. Even without PC data, it can go very well. And then after that is done, during your PPQ program, you could then do more of the PC studies. You feel more confident doing concurrent PC studies versus having it as a prerequisite for the runs. So I hope that gave you a flavor of how we do things here at Avid and process validation in general. Happy to take any questions. I thank you for your time and look forward to any questions.
Okay, I'm going to pop back on. Thank you so much for that presentation. I'm going to remind the audience that if they have not submitted a question, they can do so still. And with that, we're going to get to our first audience question. And that question is asking, what makes tech transfer one of the most critical inflection points on the path to GMP? Can you help with that question?
Oh, sure. As I said in the presentation, the PPQ program starts with a tech transfer. And I think a lot of people, a lot of people have some assumptions that the process is fine, ready to go. And then just to be honest, on the CDMO part, we'll say, okay, this process can fit into our platform. And in neither case, either is right. And so what you want to do during that tech transfer, it's so critical, is that you don't want to assume success, but you want to do a critical data agnostic type of evaluation by both teams, by the client and by, in this case, the CDMO, Avid. You look at the data in a very critical way, agnostic way, and you really do an assessment of what it takes to do a successful tech transfer. Do you have the data to support it? Yes or no? And I think the inflection is really making sure that both teams are on the same page for that tech transfer. Once that happens, it really makes you kind of clear the ground for the rest of the process. So I think that inflection point means doing that proper tech transfer and really doing a proper evaluation from one study to a next, getting the data, taking the time to evaluate the data from one study to the next before you go on. It's a kind of a stepwise approach that really needs to happen. But being deliberate at that stage pays dividends going on to the PPQ.
Perfect. Thank you so much. Now, this next question is asking or saying, hi, I'm a fellow chemical engineer at UCLA. I liked how much emphasis you put on tech transfer before PPQ. As I am interviewing for an upstream manufacturing role at Avid Bioservices, what upstream specific, I lost my place here. Let's see, parameters or process gaps tend to cause the biggest challenges when moving into PPQ? And you can see that question too, Richard.
Yeah, I don't. Well, first of all, you're in luck. I'm a chemical engineer from UCLA, so that always helps.
Here we go.
I didn't see that. I'm sorry. Can you repeat that question? I don't see it on my screen here.
Perfect. It's number 8. So this question is from Lexus, and it's asking, as I'm interviewing for an upstream manufacturing role at Avid Bioservices, what upstream specific parameters or process gaps tend to cause the biggest challenges when moving into PPQ?
Yeah, that's a great question. I think what's happening today is that these are very high performance cell cultures, and meaning that they're very, they grow very well. They produce an incredible amount of protein. Right now, as an industry, these cells, typical cell lines produce maybe 100 times more than when I was at UCLA as a chemical engineer. That's how much these cells are making, and they're very sensitive, and you have to be very careful. And so one of the things that we have to understand is how the cells react to different conditions, how they react to feeds, how they react to the nutrients, and how they react to metabolic metabolites. I mean, like CO2 and ammonia. So it's that knowledge of how robust the cell line is when they have to be so finely tuned to manufacturing, if you know what I mean. How finely tuned does the pH control need to be? How finely tuned all these need to be? Because these cell lines could be, in some cases, little bit on the finicky side. So really having that understanding of what the cells can handle at large scale.
Very good. Thank you. And thanks for that question, Lexus. Next question is asking, what is the typical duration for the tech transfer and how are ways to accelerate it? And has this been accomplished in the past?
Yeah, it's a great question. It depends on the client data package. Some are almost ready to go, but typically, just in terms of how we need to evaluate things in the PD lab, it's typically going from start to a GMP thaw within 7 months, 6 to 7 months, is pretty fast, and that's probably the fastest we can probably do at this point. To accelerate it, though, like I said, it really depends on the success of that tech transfer. It's possible if you have the data to support the small scale data to support kind of the large scale or your GMP scale manufacturing process, that accelerates it. We don't have to repeat a lot of studies if that data is available. The only thing I could say is it's possible many clients think they have the data that's acceptable for a GMP large scale, but it needs more work. And when you're dealing with cell culture and purifications, you can't fool it. If you don't have the data, if you don't have the knowledge, you're going to go into that first run at risk. And these risks and these runs are millions of dollars, and you just don't want to have that risk associated with such a program. So more knowledge, more process data in the beginning will accelerate the tech transfer program.
Okay, very good. Thank you so much. Next audience question is asking, at what stage are comparability studies performed? How about this one?
Yeah, I mean, typically, one of the case studies I gave was actually a late stage project, and typically the client will perform a comparability of the material used at Avid and the material used at their donor site. Typically, that would be done in that first GMP run, that tech transfer and GMP run. I mean, all throughout that tech transfer process, we'll send samples out from our labs to show it's the same as their donor site. We would confirm it at the GMP scale just to confirm. But during your PPQ, when you do these 3 runs at scale, those samples would also be evaluated against the donor site and really need to show that if the donor site is a commercial site and you're going to be commercial too, that these 2 proteins that we both make are the same. This is the same protein by both processes. That's typically during the PPQ program, and it's obviously something that would need to be submitted in a BLA to show that it's comparable.
Okay, very good. Thank you. I'm not sure if you covered this one yet, but the next question asks, well, great presentation. And does Avid perform DOE to identify critical process parameters and their range, or is this required from client?
Thanks. Yes. We identify the CPPs if needed, if the client hasn't, or we verify it with the client during the PC studies. So typically, PC studies require to do them very well and to do them with statistical power. You want to have high throughput systems. So at Avid, like I mentioned, we have the AMBER system. That's a system of 24 identical reactors at the 250 mil scale. And through those, you can really identify your critical process parameters. You can really verify with the client those critical process parameters. And of course, and the same thing with the downstream. We have a TCAN robotic system where it can do a number of purification ranges, purification conditions. So these DOE studies, these large-scale DOE studies, these high throughputs, these are really critical. Typically, the CPPs are identified by the client initially with Avid, but through the PC program, you can really verify those CPPs, or you may identify new CPPs.
Okay, very good. Thank you.
Mm-hmm.
And now the question is asking, Usually, how long does it take for the entire TT process to complete, especially if it's a new study with no supporting data?
Yeah, that's with no supporting data, and that's including assays, which is I haven't really touched upon that today, but it's also very critical. Um, Typically, we're looking more in that 9 to 10 to 12-month range to go from start to a completion of a GMP batch. A lot of this depends on the type of protein that we are asked to do. Typically, antibodies are they're almost like a commodity. They're very straightforward. Avid has, we can say we have a platform, or we have a toolkit, which is a more appropriate terminology. We have a toolkit of media and feeds and purification chromatography techniques for antibodies that we can go very quickly. We have a toolkit of assays that we've shown in the past works. So a lot of it is depending upon if we're allowed to use a toolkit or if there's any sort of custom. Sometimes more difficult types of proteins may require a different, a custom type of assay or a custom type of media, for example, or resin. Those take longer. But in general, let's just say 8 to 9 months for an antibody, 9 to 12 months for a non-antibody would make sense.
Thank you, Richard. Now, this question asks, based on experience, where do programs most often underestimate the complexity of scale?
I think a lot of times it's funny. It's always assuming that everything would scale. I guess in a gentle way, people think if it works at a 2-liter in their lab, it'll work at a 2,000-liter in my facility. It's just not the case. So I think the teams kind of fall in love with their own data, if you will, and they don't realize, when you scale up, especially reactors, the cell environment cannot be duplicated exactly. You can model the performance, you can model the yield and the growth, but they can't be scaled up exactly. And so just assuming things is just a bad start. And I think potentially there's a lot of a lot of thinking out there that this is very routine, and unfortunately there's a lot of just unintended consequences that through the program I just mentioned you kind of eliminate. You know about it, the process knowledge, you understand exactly what is acceptable and what is not acceptable to do for your process. So I guess at the end of the day, just misunderstanding that yes, you can scale up. Yes, there's mathematical ways to do it. Yes, there's ways to do it, but there is no guarantee about any sort of unintended consequence of scaling up. It's not just something you can just do as a paper exercise. You have to really experimentally determine this.
Okay, very good. Thank you. Now, this next audience question asks, what are typical CQAs and CPPs that are most critical to the success of biologics?
Well, it depends on the assay and the type of protein. Typically, the most critical CPPs are probably in the upstream, with request to the glycosylation of your protein. So your protein has sugars on it that affects its half-life in the patient, for example. Where does that come from? Well, that comes from the cell culture conditions. And so that CPP of cell culture conditions, it could be a pH. If you really have there's projects at Avid that we had to make the pH control extremely tight to make sure that those carbohydrate profiles were the same as expected. The other CPP is typically high molecular weight species from your protein, essentially. So when you do purification, you want to make sure that when you collect off your columns, that you don't collect areas from the chromatogram that could contain high and low molecular weight species. You really want to make sure that your collection criteria is acceptable and reproducible. And the last one is some of the process-related impurities like host cell proteins. For non-antibody type projects, those host cell proteins are a little bit trickier to remove in some cases. And so you have to make sure that even though your product may be high purity, the contaminants from the host cell proteins may be high, and you have to really identify where in that chromatogram those are coming and making sure that's not part of your collection criteria. So those are the three major ones that people are concerned about.
Okay, wonderful. Great questions coming in. This next one is asking, is tech transfer, scale, and site changes for biotherapeutics before BLA submission and at the commercial stage?
They're both.
Something.
Yeah, I mean, we've onboarded both prior to commercial and after commercial. So after commercial means that the client has a commercial place set up either internally or another CDMO, and they want a second source. So again, at that stage, that is typically a little bit easier in some ways because it's already been through all the steps I showed, the process characterization at small scale, process performance at large scale. Typically, they would transfer a process to us that is at the same scale. The 2,000 liters that we use is really an industry standard at this point. And so most likely, if they're at a 2,000 liter reactor, we can onboard that commercial process very seamlessly. We don't have to revalidate as much. We have to show process performance. We have to do a PPQ protocol. We have to show product comparability between their current site and our site. But it is actually, in some ways, easier to transfer a commercial project to a CDMO versus a pre-commercial project, which majority is pre-commercial, but is actually the activities can happen at each stage.
Good, thank you. And this next question asks, what distinguishes PPQ-ready processes from those that are simply transferred?
Yeah, again, the PPQ-ready process is a process that has been performed many times before it comes to Avid or a CDMO that's ready to go to PPQ. Yeah, I think a PPQ, something that's ready for PPQ, you have that process history of performance, batch after batch of being reproducible. Now you have something that's ready for PPQ. What you also need at that same time to be ready for PPQ is making sure, again, there has to be some small scale challenge to your process to make sure that your limits are reasonable and that your batch records that you're going to be executing are within those limits. So again, process knowledge and reproducibility at GMP scale is really a good foundation to have before you're ready, before you're ready to go into the PPQ program.
Richard, I think we have time for one more question.
Okay.
I'm gonna ask, well, this person would like to let you know, very good discussion, was interested to know how contamination control strategies factored into tech transfer.
Yeah, it's, of course, contamination control strategy is a corporate strategy. So at Avid, we have a policy of a contamination control. And we have policies to show how we handle the process is acceptable, is low risk. By being disposable, there's really, very little chance of bioburden and endotoxin. We get that, but your procedures have to be robust to ensure that you have bioburden control. At the end of the day, what we do for client projects is just to make sure that the raw materials that they use for our process are acceptable. So cell banks that are delivered to us, they have to pass strict testing requirements for viruses and mycoplasma and sterility. Any raw materials that could be unique to a client has to make sure it comes from a qualified vendor. It has to make sure there's no animal source materials in it. It has to make sure that it has the proper C of A's, quality attributes to that material to make sure it's safe. So at the end of the day, it's not the major part of a PPQ, but it is a checkbox that we have to make sure that this program that we're executing under PPQs falls under our contamination control strategy.
Very good. Well, that is our time for today. I want to thank our speaker so much for taking the time to share his information and experience, and thank you, the audience, for watching. Just a reminder that this presentation will be available on demand. You'll receive a link via email as soon as it's available. Have a wonderful day, and we'll see you next time on Outsourced Pharma. Goodbye.
Thank you.
Thanks, Richard.
Bye-bye.